Hi, everyone. Really nice to see all of you. My name's Joe Lally. I'm a PhD student in Donald Whirling's lab, and I'm here today to introduce Mike Love, who is a biostatistician out of University of North Carolina. Um, I can't think of anyone who better embodies the spirit of open science than Mike. Um, his whole career, he's been focused not only on understanding the statistics behind RNA sequencing and ways to actually extract viable information from oftentimes noisy data. But he has, throughout his career, worked to ensure that others in the field can understand and access his research. One of his most cited papers is actually just a how-to on how to use his tools. And I, as a PhD student myself, that's how I first encountered Mike's work, is through these online tutorials. And I'm sure that there are other PhD students in the room and professors in the room who learned how to do RNA-seq analysis by reading his work. As a result, the tools that he's developed have become standard in the field, such as DSeq2. And in addition, that kind of like open-hearted spirit of allowing others to, encouraging others to use his tools, and properly examining the limitations, and you working with other people to ensure that when there's a better tool that comes along, he would encourage others to work with those as well, has ensured that he stayed in the forefront of this field, because everybody loves to work with Mike, because Mike loves to work with everybody. This talk started as a GitHub issue. We were discussing last night, actually. I was reaching out to Mike about one of his newer tools related to this topic of allele expression, Fishpond, and different ways that I might be able to use it in my research. And we ended up meeting over Zoom, and then there was a request for potential speakers, and I thought, well, Mike would be a wonderful speaker. And no pressure now. I'm a great admirer. I'm very happy that Mike is here. And without further ado, here's Dr. Love.
Thanks so much, Joe, and for that very kind introduction. That's what I would hope, if I can accomplish, is like encourage people to try things out and be open about your computational work. And Madison's such a fun place for me to visit. I was very excited to accept, and I get to chat with lots of friendly people who I've known for a long time. So this is all the students here, you're in the right spot. This is a fantastic place to do statistics and genetics and data science. So I'd like to talk to you today about something I've gotten interested in, somewhat recently in my career. So I didn't think much about the alleles that I was looking at, through my PhD, into my postdoc. I was just looking at, functional genomics data without really considering the fact that there are two alleles in the, present that are generating these sequences. And then I really got interested in it finally when I ended up at UNC, because, so if you don't know, UNC Genetics does a lot of mouse work with mouse and work with a set of experiments called the collaborative cross. So they have a lot of F1 mice, and they were asking, like, well, can we use these tools to study allelic expression? So it's kind of a winding path, like a Dora the Explorer path here. So first, I'm going to talk about how to compare gene expression in cases where we have a lot of measurement uncertainty with these short reads, so short read RNA-seq. And then that, being interested in that question then kind of led us to people who were saying, well, can we compare across alleles with short read RNA-seq? Because those, you have a lot of uncertainty because alleles are very similar in sequence. And then that led us to ask, well, can we also do this alleles and isoforms at the same time? So when we have heterozygotes, can we see differential signal at the level of isoforms? It's like, maybe, I think will be the answer a little bit. And then the final conclusion is like, it will be much better when we have long reads. So that's kind of where I'd like to continue taking this work. So, sometimes it helps me to gauge an audience. So from the people in the room, who has done any kind of like allelic work where they ask about the two alleles, which is fine. So, okay. So this is like me, I was like, brought into this field. Actually, I think the person who really got me interested, aside from the UNC folks who were asking me, was I was teaching with my postdoc advisor, Rafa, at Cold Spring Harbor, and Tuuli Lapalainen, who was giving lectures as well. It was also an instructor. And she was talking, I have some slides from her recent papers, and she was just, kept saying, like, this is a very important signal. It's really worthwhile to spend the time to try to extract the signal from your data. So that was something that kind of, I would always think like, yeah, I should maybe try to incorporate this. So I'm going to start off on the left side, we were trying to compare gene expression when there's a lot of measurement uncertainty. So, we're interested in performing, the motivation, I'm not going to start from like why we care about RNA-seq. I think this group probably, has heard of RNA sequencing and understands why we would be interested in measuring the abundance of RNA molecules. So we want to perform analysis at the best resolution possible. And so there's a slide I'm just stealing from a statistician, Susan Holmes, where she quotes Don here, premature optimization is the root of all evil in coding. And then Susan twists it to premature summarization is the root of all evil in statistics. So if summarization is incredibly powerful because you can, it both reduces the data, it might increase the power because if you have random errors, they might collapse away and you have better signal. But if you're missing the right signal at the right resolution, then you've lost it. And so kind of figuring out when to go higher in the aggregation and when not to is like still a really interesting question, I think. So I'm going to use this data set for a couple slides. So this is a data set from a paper from Kaur Alasu from Dan Gaffney's lab. They did macrophage from a bunch of different human donors. And then these are, I think, these are iPSC-derived macrophages. And then they stimulated them with interferon gamma, among other treatments. So this is like a nice little data set with a treatment. And it also has a paired design because they're from different donors. This is just a subset of the data. So the blue, you have two pairs, two boxes, which is a single donor, and you can see like if it goes up or down. And then here, the box is actually, so we don't always know the count for a gene. This is a particular gene I'm showing you, and we don't always exactly know the count if some of the reads map across the genome. And so like Colin Dewey here kind of, really, set the standard for how best practices for coming up with a count when you don't actually observe it. So you can use expectation maximization to distribute those. And so this is kind of like that, the box I'm showing you is like the uncertainty that we don't exactly get a count, but there might be some uncertainty associated with that. And so, this is like a rough sketch of what's happening. The cDNA were shattered into smaller fragments. Those, before this, there was actually reverse transcription. And then we sequenced these reads, maybe with a pair, and then we map them. And then maybe we assign them probabilistically. And so in this case, we're using, Salmon, which is another EM type algorithm. So that's what's on this like probabilistic assignment of reads to genes is what's on the y-axis here. And just looking at the pattern, there doesn't seem to be a consistent pattern at the gene level. But then if you zoom into the transcript level, let's see, maybe I'll use, if I use the pointer, then the people online can't see, but I'll describe it in words. So there does seem to be a pattern at the transcript level, right? So the first this 648983, which is this bottom one, it seems to be kind of consistently up regulated by interferon gamma, and this other one seems to be down. So they're doing some kind of switching, and you don't, because they cancel each other out, you miss it if you just look at the gene level. And so, you know, we had done a lot of, I and some collaborators like Charlotte Sonneson and Rob Patro, Mark Robinson, we'd written papers like, this is how we can use transcript level estimates to do gene level analysis. And then we got interested in, well, let's also try to see what methods there are for working at the transcript level. So we have to, at the isoform or transcript level, we have to deal with the uncertainty. And so I like to use an analogy of like a puzzle, right? So a read is like a puzzle piece from this big puzzle, which is the transcriptome. And there's kind of two types of, there's two types of ways that it could be ambiguous. And this is kind of like within a gene, two isoforms share a lot of sequence. So there's ambiguity there, or there could be like two regions in the genome. So there could be genomic multi-mapping. And so in either case, we have to, account for that. So both at the gene level or much more at the isoform level. There are a lot of methods to deal with uncertainty when doing, when performing differential expression. So there was like MCMC based methods, some kind of over dispersed negative binomial mixture models, bootstrapping from Madison group from Christina's lab, EBSeq, and I have a slide about that. Taking into account the over dispersion from uncertainty related to isoform number, a Bayesian model that had random effects, another bootstrap model, and then just recently, this is like from December, there's Gordon Smythe's group has come back and tried to incorporate it into EdgeR as a weighting term. So I'll come back to the last one also. So I like this paper. It's very clear that the measurement uncertainty is visible stratifying the transcripts by, number of isoforms per gene. So you can see, genes that have more genes that have many splice isoforms grouped by, how the complexity, you can see they have differential variance, like marginal variance. Just looking at the variance across samples, they have more, and that's makes perfect sense given that there's additional uncertainty in the assignment of the reads when there's a lot of when there's multiple spliced isoforms. So that, treating those kind of as stratifying the analysis and treating them separately shows an improvement. So we were, interested in, this is when we were doing some benchmarking. This is like before we thought to try to develop any methods in the space. And we had built a model for bias. So, the fact that coverage is not uniform along transcripts, but it goes up and down frequently. So we built a model for bias that we called Alpine. And then that is important because the optimal case is that the coverage is uniform, that reads are drawn uniformly from every exon proportional to their length. That would be the best case scenario. It doesn't happen in practice. Often there's coverage dips, which just makes the problem worse. So we simulated reads with empirical biases, basically like throwing out reads that we were unlikely to see in real data sets. So for example, fragments, reads originating from fragments with high or low GC content are less abundant than in the middle. And we ran different methods and then compared. So here the circles are the 1%, 5%, 10%. And we look to see the sensitivity on the y-axis over the false discovery rate. And methods were kind of, so this is the first slide I showed you was 3, 6, 9, 12. And, there were kind of groups of methods. There was, cluster of methods here. These ones, I think they were lower because of filtering choices. So they were, at the first step, they like filter out a bunch of genes, or transcripts in this case. And then there were, this sleuth method, as I think it has a problem with consistency of estimation of certain parameters. So that's bad. If you have an inconsistent estimator, as your sample size grows, the problem gets worse. So we thought, we were actually thinking maybe we could, throw another method in the mix here. And I know this is actually pretty blurry, but this red one, seeming to hit the mark pretty well, was called SAMseq. And so SAMseq is just it's a non-parametric method that involves resampling. And we thought that might be a promising method in this space. So we first sought to quantify this uncertainty. So like this kind of mapping ambiguity here at the level of the transcripts. And I'll just mention this is work from Anshi Zhu, who was my first UNC Biostat student, and now she's at Genentech. So we measured this. So if you look at the box as a single sample, and we measured the sample variance across multiple realizations of the data. So basically, we have a model for the data, and then we can estimate, how many counts for this feature, for this isoform. And then we can, there's one of two ways that we have to generate other possible values. One is bootstrapping, and another was a Gibbs sampler that was derived from a previous group, Ernst Turo. And so these actually give kind of similar estimates of the variance, of the inferential variance. So if we were to, get another plausible value for the estimated count for that feature. And so that's what's shown in the box plot. And then the problem is that variance is not a great thing to compare across transcripts because it depends on the mean. And so minimally, you have kind of Poisson variance. So that's what we do here. We minus the Poisson variance that's expected, and then that would still trend up over different transcripts by their mean. So then we divide by the mean. And then this quantity, which we call the inferential relative variance, is flat across transcripts by their mean. So if you compute this, and this is like a very obvious plot to make, it's lower for gene level estimates than for transcript level estimates. And over here, this is probably like, these are going to be likely the groups that EBSeq was stratifying by. Like there's high complexity genes that are very difficult to quantify at the isoform level. And there's others where it's okay. And then there's like at the tail end, there's genes that have a single isoform. So then we kind of just built a method on top of SAMseq. And so SAMseq, the idea behind SAMseq, the really quick high-level idea is samples differ by their sequencing depth. And you can think of the data. So here, this is a little swap. Here is the sample. So usually we look at tall gene expression matrices. This is wide. So your counts that you get are it says, well, they're kind of like a realization of a Poisson random variable, and sequencing depth is just a scaling factor. So if new IJ is the expression level, that goes up and down by this unknown DI. We can estimate DI, and then we can create new data by just sampling. So if we estimate all the DI, we can just downscale them all to the lowest sequence sample. And then that's one realization, that's one like Poisson resampling, and then we can work within prime rather than within. And then they note that, well, that kind of solves the sequencing depth issue. It does lead to a loss of information and inefficiency if D min is very, very small. So then instead of doing this downsampling, they kind of downsample some and upsample the others. So you meet in the middle, you get a new count data set from the Poisson sampling. And then what they do is they just do some non-parametric statistic. So if you're looking for a difference, if you're looking for a shift in the location, you could do a Wilcoxon. And then obviously you can generate as many of these n prime matrices as you want. So they just say generate a bunch of them and take the average. So to average over the Poisson variation. And then, yeah, so that's what I just said. And kind of the intuition behind this is like if you think about from the perspective of a Wilcoxon test, these two data sets have the same Wilcoxon statistic. There is separation between the two groups, but one of them has much higher count. So when you do the resampling process, you break the pattern many times on the left, whereas on the right, the Poisson variation doesn't, break the separation. So that's kind of an intuition how this averaging over many Poisson realizations is useful for RNA-seq. If you've ever looked at RNA-seq, like, well, those are too small, and those are better. They have less, variation, and at least on the log scale when you're trying to compute log fold changes. So we called our method SAMseq with inferential samples helps. We made it very clear that we're not like devising a totally new method. We just kind of, what we did was we take our data sets are not Poisson resampled data sets, but they're from the bootstrapping of the reads and then doing the EM. So that has the uncertainty baked into it from the read to transcript assignment problem. We average the statistics, and then we do permutation for p-values and q-values. So because of this, it doesn't work well when the sample size is very small. But if you have like 5 samples per group, this seems to work pretty well. And we then like extended it to some other designs that weren't included in the original SAMseq paper, with like thinking through how you would do the permutations to test for other things. And so that worked well in these in this benchmark that we generated already for this other paper. It seemed to control. And then what I'm showing here, this is like 6 versus 6. This is 12 versus 12. This is the overall plot. And you'll notice like all the methods are doing pretty well here and comparably. These next 3 plots are when you stratify the transcripts by the uncertainty. So the lowest third, middle third, and highest third of uncertainty as measured by bootstrapping or Gibbs sampling. And so just the point here is like this method, because the way we're doing this averaging is controlling better than these other methods in this one simulation. We also ran it on some null data sets to see that it would do okay in controlling false positive rate. And then I want to mention that there's this new method, which also appears to be really good from the EdgeR group. So it's this method called, in the paper, it's called EdgeR scaled, where they're doing, it's similar to what they do in Lemma-Voom, where they scale based on precision. And so here, knowing that there's a lot of uncertainty in the read assignment to transcripts, they pass that along to the GLM by scaling down the count. And that seems to work very well. And the benefit there is that it can also work when the sample size is small. Whereas our method, because it relies on permutation, will suffer at the low sample size end. So that was our first foray into dealing with a lot of uncertainty in the measurements. In this, like, we have a count matrix, but it's not exactly an observed count matrix. And then we did some other projects. So a student from the lab, Scott, worked on, is there uncertainty in single cell? Yes, there is. It has to do with the fact that you only sequence the 3' end. So there was multi-mapping we could observe across genes because the reduced representation, not many people seem to be worrying about that the matrices are so big, they just want to have a count matrix. So like, I don't think we got much, we didn't get much out of this, like telling people to worry about the uncertainty of gene multi-mapping in single cell. But if you try to go then to isoform level, it would be non-ignorable. And then I'm going to talk more about this second project from Yufi Wu, who looked at trying to do allelic imbalance at the isoform level. And then the last project, which is by Noor Singh at UMD, so with Rob Patro's group. I think this is very exciting, and this is an ongoing work. He has one publication and one preprint in this space, but I think there's a lot of space for the whole field to move in this direction, which is data-driven aggregation of transcripts to kind of optimally find the resolution for doing differential comparison. So I don't think I talk more about it, but like this is something that we're still working on. So I want to talk about alleles now. So that's differential expression when the features are very similar. So they might differ just by a single nucleotide. If they don't differ by a nucleotide, we can't do anything. But if there's an exonic variant and the donor is heterozygous, we have this extra information that we can use, which is very interesting. So I was told I shouldn't use the letters AI because that's in use. Yeah, that refers to something else. So I'm using allelic expression imbalance, AEI. And so why is this interesting? So it's a reliable and robust measure of cisgenetic regulation. So if you're looking at an organism that is heterozygous for a non-coding piece of DNA that in one way regulates the gene, and that I'm showing you, this picture is one mechanism. There's actually lots of mechanisms that could be relevant. So so one mechanism is like it affects the binding of transcription factors to regulatory elements. And so you can observe this if a couple things are present. So if the transcription factors are present, I've drawn a couple of like colorful little transcription factors, and if the regulatory element is accessible, you will see differences between the two alleles. It's like an integration of all the potential regulatory elements affecting the transcription of the two alleles. And so because it depends on the abundance of the proteins and it depends on the chromatin, it's very context specific. And so if you care about this variant in particular and you want to know in which cell, in which time point, in which condition is it active, allelic imbalance is a really good readout. And another thing that's exciting about it is that it is less affected by technical artifacts because well, if there was a technical artifact, let's say PCR induced an imbalance in the way that the RNA transcripts were amplified, these sequences are very similar. So they have nearly identical GC content. They have they probably have nearly identical length. So there's many ways in which the two sequences are very comparable. So if the bias is systematic and depends on things like length or sequence or structure, secondary structure, these are probably very similar. And so the bias affects them both in the same factor. So it divides out when you compare the two. And a lot of biases are multiplicative. So that's another, so it's like you have an internal control for this experiment. So you get a fold change that's pretty robust to, batch effects, technical artifacts, and also just transgenetic effects. So, effects from the other part of the genome or environmental stimulus on the cells, like that's all, you just get the cisgenetic readout. So that what you need is the donor has to be heterozygous for the regulatory variant, and you also have to observe the allelic difference. So you have to also have a heterozygous variant in the exonic sequence. So this is one picture. There's also post-transcriptional regulation and you could have like a variant that directly affects splicing. So it could lead to like nonsense mediated decay, or localization of the transcript as well. So it's very exciting to do this in single cells. So there's a bunch of methods that have been proposed. This is just a short list, and I stopped making this slide a couple of years ago, so I'm probably leaving out important methods. But there were a number of methods that had already looked at this in spatial and single cell data. And so you can see cool patterns like here. You see differences in the allelic readout, depending on where you are, where these cells are, either in the state space or in a spatial region. So we had one paper on the single cell space where we said, if we have cells that they've already been grouped into cell types, maybe by their total count or by some extra information, maybe the cell surface markers, can we robustly look for shifts in the allelic ratio? So we used a fused lasso to kind of see if we could, if the ratios are similar, the fused lasso should reduce them and put them in the same bucket, basically. And so this was like pretty simple project. It seemed to work very well. The signal is really sparse. You have a lot of, very low counts, and you're looking at these ratios. And so it works. It's important here that it works on the deviance, which takes into account the high variability when the counts are low. So that works well to you just want to quickly group the cells that have similar allelic ratio. And we have a cool, all the R packages have to have a cool hexagonal logo. And then another project, this is an applied project I worked on with a group in Berlin, where Anna here did single cell RNA-seq and ATAC-seq of an F1 cross of melanogaster and Seychellia. These are very divergent Drosophila species. So there's a lot of work involved in genomes, and that was something Anna wasn't expecting, to spend so much time working on patching genomes and figuring out where gene models fall. But then we used another method within the salmon ecosystem called ALVIN to assign reads to genes, do the bootstrapping to account for multi-mapping across the genome. And you can end up with these clusters of cells, and then you can look at the allelic imbalance between. So how have the species diverged in their cisgenetic regulation of genes? And how does that change across the embryonic layers? So there was, this was such a fun experiment. The Seychellia, one of the things that distinguishes it the most from melanogaster is it is able to eat this toxic fruit that grows on the island where the Seychellia Drosophila live. And so when you looked at the allelic imbalance, we had olfactory receptors as the most differing things, which was, that is the positive selective sweep that allowed this species to survive. So that was, reassuring. We then worked with a lot of collaborators. We've looked at allelic imbalance across human neural progenitors and differentiated neurons. This is showing you that the signal is similar within heterozygotes and comparing across homozygotes and heterozygotes in an eQTL effect size. So that you would hope that it would be a similar effect, except for trans eQTL would not show up in the allelic imbalance. If you're interested in toxicology, there's a really cool paper from Francesca Luca's lab over at Wayne State, where they took 6 donors, lymphoblast cell line, created iPSC lines, and then iPSC-derived cardiomyocytes. So you have 6 donors, 3 cell types, and then they expose these cells to 30 different chemical treatments. And so this is getting back to allelic expression as this nice reporter of something going on. So if there's a G by E effect, you might only see the allelic imbalance occur when you treat the cells with nicotine or with caffeine or with, lithium. So in my opinion, it's actually a pretty cheap assay to see a G/E effect. You just need to have a heterozygote. You need to have the heterozygote at the regulatory element and at the and within the exons, but that actually occurs enough that this is a reasonable design. We've done this also in adipocytes at UNC, and yeah, I mentioned the Drosophila work already. So I said in the title, like, this has some disease relevance, so I want to point to that. So there's a nice paper recently from Mohammadi, and this is from Tuuli Lapalainen's lab. So they looked across GTEx at allelic expression imbalance. So you have a per donor imbalance for every gene. And then they quantified how much allelic imbalance is there for this gene when we look across a population. So if there's a lot of variance, if there's a lot of allelic expression imbalance, that indicates that evolution has permitted a lot of cisgenetic variation on the dosage of this gene in this tissue. Okay, right. So if it was not allowed, if the gene expression dosage it would be a bad idea to have it varying a lot. There would be purifying selection to avoid that, to remove that genetic variation. And then so they compute this. It's basically like, you can think about it as how much cisgenetic variation affects allelic imbalance. And you can look across these categories and their color. So the colors are like whether it's significantly different than randomly sampled genes. So these are particular subsets of genes, and then this is like the average. And the ones where the variation is lower are like mutated in autism, mutated in different diseases, haploinsufficient, loss of frequency intolerant, dosage sensitive. Yeah, and then the ones on the right-hand side that seem to be higher are loss of frequency tolerant, non-essential is the last one. So it does seem to be an effective measure of, how tightly controlled, how much does it matter that the gene expression varies or not, according to cisgenetics. That was just standard deviation. So that's an aggregate measure across all the donors. They also looked at outliers. So when, one individual has a really deviant change between the two alleles, and they looked at genes where that occurs, and they see if you try to predict genes that have these outliers, they are often variants that are low frequency, and they are associated with these stop gains, frame shifts, and splice site genetic variants. So this is kind of expected, right? Like, you would expect these to be rare because they lead to a big cisgenetic change in the expression, and they are of the type that you would expect. They're kind of pretty big effect variants. These are, I should say, right? When we see allelic imbalance, we don't know the regulatory variant. So this is looking, this is an aggregate statistic looking if these types of variants are nearby. So that's a pretty important point. We can't, with a single donor, we can't say this is the regulatory variant because they will be heterozygous for a bunch of variants nearby. So the only way you can do this is by asking, like, how often are these types of variants nearby? And so you get a kind of enrichment rather than being able to pinpoint. So then in the back half of the talk, I'm gonna quickly go over, we can try to do this at the isoform level, but it's very difficult. So I've shown you all gene level up until now, allelic imbalance. And so there could, if there are multiple regulatory elements, and they're each kind of paired with different promoters in this picture, you would get differential, you get this allelic imbalance, and it would be differential across the different isoforms. And so we were wondering, is this pattern there, and can we observe it with short reads? So it was, there had been people who had tried to do this. So Gary Churchill had written a paper attempting to, it's basically like RSEM, but aligning the reads based on hierarchical rules, whether it's isoform disambiguity or allelic ambiguity. Right, so there's these different types of ambiguity, and there are a lot of reads that are isoform and allelic ambiguous, right? Which is like these two types. But there are some which are only ambiguous about alleles, and there are some which are only ambiguous about isoforms, and you can use EM to try to do your best to estimate both isoforms and alleles. So we went with, we tried to kind of, we actually didn't use their hierarchical approach. And then we tried to see, what can we do? How does this method perform? In this case, I was working with Rob Patro, so we're working like downstream of the salmon quantifier. And this is work with KB Choi and Gary Churchill. So first, I just want to say, like, this is all a short read task. The problem is pretty much solved if you use long reads and you have enough depth. So like papers are now coming out that, like, this is just from just published like in August 2022. You can find these very nicely in long read sequencing data. So like this is alt and ref, allele, and these are two different isoforms, and they're doing this switch. So you just totally miss this signal if you summarize directly to the gene level. Just counted up the reads in the locus, you would totally miss the fact that there's a cisgenetic switch. And this was just a, look, there's only 5, there's only 5 donors here that were profiled across tissue to look at tissue-specific allelic transcript usage. But even still, they were able to like do these kind of enrichment analyses, and they see kind of what you would expect. There are rare heterozygous variants nearby. They're enriched. They fall into categories that you would expect would cause AEI. So we were trying to do it with short reads. So you just have to get lucky that the short read. So these are, and you can't really see this is an IGV plot. So there's a alt allele right there. So we have to get a paired in read where we get something overlapping the heterozygous site and a part of either that read or its pair giving us some information about isoforms. And then we do, again, like either bootstrapping or Gibbs sampling to estimate all this uncertainty across all these different categories. So we developed a pipeline. So basically, the first thing you do is you build 2 copies for every organism in your data set. So you give it the 2 possible alternatives. There's a tool from Gary Churchill's group to do this. You just run salmon or RSEM or Callisto to give it the 2 options, the 2 alleles for every isoform. And we built some software to then easily import this into R, given that we particularly care about pairs. For allelic imbalance, we eventually are going to want to compare the 2 alleles. And you can do it at different resolution. So you can do it at the gene level. You could group isoforms by their promoter, or you could try to do it at every isoform. And then we use this tool again, which accounts for all this uncertainty to try to do differential analysis and make some plots. So I'll finish by showing some. I'm not going to show you simulation. I'll just show you straight to the data. So this is, we had a nice F1 cross. Like this is a very classic cross of cast and black 6 mice. So there's a lot of heterozygous sites with this cross, and this was osteoblast precursors differentiated into mature osteoblasts in a 9 time point time series, collaboration with Cheryl Ackert Bicknell at Colorado. So here's these are 2 transcripts of the same gene. So transcript 1, transcript 2, and this is the time course, and these are the 2 alleles. So this is a case where like one strain prefers one allele and the other strain prefers the other allele. And we try, it's pretty multi-dimensional data. So you have to kind of look across a lot of different plots to figure out what's going on. You might also be interested in the total proportion of the 2 isoforms, like which is more highly expressed. So, we tried this, and what we saw was actually the majority of the genes, all the isoforms, if they had allelic imbalance, it was consistent. It was not the crossing pattern. This is like quadrant 2 and quadrant 4, which are the discordant cases. So most of the data is in quadrant 1 and quadrant 3, like this. So these are the other quadrants, and we use this to peer into, are there genes that have high counts where we see this discordant signal? And we looked at all of these. And so here's some examples of where we see different patterns within the same gene. So these are kind of the dynamics is interesting over time. And another thing to think about is there could be biases due to our inability to have a great set of reference transcripts, but those would probably not be dynamic patterns, right? If there was a reference mapping bias, it wouldn't be fluctuating over time. So if we see a fluctuating pattern, we're more confident that it's maybe due to cisgenetic regulation. So this was a work that we published last year, and there's some software. I think really the answer though is new data, as happens a lot in genomics. New data comes along and then old problems are solved, and that's fine. So this is my very last slide is you can do great things with this new data. So this is a paper, a preprint from last year from a group at UVA. They had splice QTL that they discovered that are associated with genes in, again, osteoblasts. So these are now human osteoblasts. So this is a splice QTL here for this gene that clearly has a role in osteoblast. It's seen in osteocarcinoma, so it probably has to do, or osteosarcoma, so it probably has to do with proliferation, differentiation. And then this is a GWAS for bone mineral density. Bone mineral density lines up very well with osteoblast gene expression QTLs, as you would expect. And it's a very nice phenotype that you can measure very accurately with a lot of people. And so, but the problem when you do splice QTL is you're looking to see if genotype associates with the coverage of junctions, but junctions are not biological quantities. Junctions don't exist in the world. Transcripts exist. So it might be ambiguous, this junction, which isoform is implicated and what's the protein consequence of that. And so long reads are very useful then you can project your discoveries, like your splice QTLs, with long read data, and that's what they've done here. So they're like, well, this light blue isoform looks really relevant, and probably this splice QTL that we observed probably has to do, this would be a good target to go prioritize for downstream functional validation. Because look, it's changing dynamically over the differentiation time course. So thanks for your attention. I want to acknowledge all the people. This is a long story with lots of grad students involved and lots of collaborators. So Anshi was the primary developer of this this non-parametric method for testing, and then Yuffie, Noor, and Scott have been like figuring out what we can do with it. This was the single cell project, and then these are the collaborators for the osteoblast data set. So I'll happy to take any questions. Yeah.
So a lot of the data you showed was in organisms where alternative splicing and introns are a lot more common. So have you seen any work, any papers where maybe like unicellular organisms where introns or alternative splicing is less common, whether or not allelic imbalance is occurring in that at relative disease with alternatives?
I haven't, but I would expect it's just like a much easier problem. So you could get by with short reads, and just looking at like stacked coverage. Yeah, so like it's an interesting signal if you have genetic variation in your population. It's an interesting signal that's like just there to go look for. Yeah. Oh, and sorry, I should repeat the question. So the question was, is it possible to look for allelic imbalance in unicellular organisms or organisms that don't have the splicing? And yeah, you could skip all this stuff about the isoform ambiguity, because now you just, you would still have to do with like gene duplicates, but aside from that, you're looking at just stacked coverage of short reads. Yep.
The 90% concordance between the gene and the isoform level, do you have any kind of additional information or expectations about whether that's going to be consistent across cell types or across species, or how variable it is, or what kind of genes might be more likely to be discordant than others?
Great question. It was actually 97% just in that, but that's just that data set. So we saw very few like of these cross patterns which is what, we were hoping there would be a lot, and then there weren't. So whatever, we let the data tell us the truth. And we were just like, well, this method would work if they existed. And this paper from the like a lot of the GTEx authors, where they did long read looking for allelic imbalance on transcript usage, they say that it appears to be not switching of the isoforms, but more to do with stability. And so you don't see the switching pattern as much. But if the regulatory variant were a splice site, you would, I think, see the switch. It's just that when they looked globally, the more common thing was a genetic variant that maybe affects the stability of the transcript somehow. Maybe it's like in the exons, and maybe it has to do with nonsense mediated decay, or it could do to like localization of the transcript or something. So they didn't see as much of the like switching. Yeah. Yeah.
We like this talk. So you talked earlier about this is a problem you're trying to deal with, the uncertainty of aligning reads to very similar transcripts when those reads are short and the transcripts are long. The transcripts and the long reads help to solve this problem, because now you can able to capture the full width of the transcripts diversity, so it makes mapping much easier. One of the challenges when trying to think about the best way to handle the problem of short reads, that there aren't a lot of ground truths in RNA-seq. But it occurs to me that long reads can provide a sort of ground truth for then trying to determine whether or not your methods are mimicking or accurately measuring the biology of the system they're trying to study. So could you take, and has anyone done this, to take the long reads and artificially split them up into small reads.
Yeah.
And see whether or not your method is able to capture the variability of these long read data sets.
Yeah, I think Rob just did that. So Rob, like a couple of weeks ago, he had a preprint called Oarfish, which was like new models for long read. And he did something like this where like the long read was he was kind of comparing across short and long. And I think he did a thing where he shattered long reads. I mean, I think, yeah, the combination of the two, and also like Mark asked me this last night, like what would be the optimal balance to get the best quantification? Because the long read I'm not certain that it gives you, I don't know yet, like as you go down to lowly expressed transcripts, the representation that's there, or as you compare across transcripts of different length category, like really short to really long, I'm not confident that it's like, I just don't know yet if it's like as good. But I think you get a good catalog of like these are expressed. This is the set that's there, and then certainly like filtering your transcriptome and just helping out the EM by saying like, well, these are the ones you could put a lot of weight then on the ones that you saw in long read, put the prior on those. That would help out a lot with the assignment of the short reads. The balance, I don't know, like that's an interesting problem to work on. Is like, should we do mostly short or mostly long, or like what are these two technologies? How do they give us together a lot of information? If there's, one thing is like if there's events, and we were talking about this earlier, is like if there's events at up two ends of the transcript, you really need long reads to tie those together. So that's, that's why you would want some if you wanted to compare splice events or alleles that are on different sides of the transcript. Yeah.
So going to the first part of your talk about using the differential uncertainty and the differential transcript usage.
Yeah.
As you know, like there's this other camp of people that have methods that operate at like the junction level or exon level, like lead cutter or XSeq and MintJulep. I'm wondering how, so they sort of bypass the uncertainty, as well as like uncertainty in which gene structures exist. So have there been done sort of direct comparisons between the two paradigms?
That's a good question. I mean, it's hard because my like biased opinion is that they're kicking the problem down the road because like, what if there's two events and we want to aggregate the information? You've now summarized at this level, and it's different. And also the other thing I think is really important is like, it's not just splice junctions, like UTRs matter. So I'm like, let's do it all in one go. Try to get the differential UTRs at the same time as we're getting the internal junctions. But like, I think that so, it would, to compare across them, you'd have to decide like what's the objective. If it's just summarizing a splice event, I would go with those. If somebody came to me and said, I really care about this junction, I don't care about knowing about the transcript at all, I just care about the junction. It's, there's a lot of advantages, like they get rid of some uncertainty, and they also, they don't require everything to be known. They don't require even the junctions to be known. They could discover them. So I don't know about like in terms of metrics who's better, but I think if you have different goals in mind, you would go different directions. And there's a lot of like, there's a paper from Alejandro Reyes and Wolfgang Huber from my PhD time that shows that the most dominant differential transcript usage across tissues is UTRs. It was not splice junctions, which is not to say splicing isn't important. It's just like there's more of this other type. There's a lot of this other type of variation that has a lot to do with genetic regulation of gene expression. So I just don't want to leave that on the table, and we can get this all in one go. It seems convenient.